PCR in medicine or in medical sciences. So PCR, it also finds its application to detect the different causal agent of diseases from clinical samples. So it can be used in clinical microbiology to know that how the infection is there and what type of microbes they are involved. So in case of clinical microbiology, the microbes, they can be studied nowadays directly by using the sample, or the microbes, they can be grown in the laboratory under certain cultural conditions, so that we can get the culture of microbes, and then the DNA, it can be isolated, and then by using microbe specific primer, the presence of microbes, it can be detected. So microbial identification is there by using the PCR techniques. So different type of bacterial causal agents, they can be diagnosed, like the pathogens of TB, that is Mycobacterium tuberculosis, infections caused by other microbes like the Bacillus, Bordetella, Chlamydia, Salmonella, Shigella. So these are different type of microbes that can cause the diseases. इसके अलावा कुछ वायरस होते हैं जो कि डिजीज़ कॉज करते हैं, लाइक हेपेटाइटिस वायरस और द HIV viruses. Their presence, it can also be detected by using PCR. For example, in case of HIV virus, although the symptoms appear, then one can proceed for the detection of the virus by using some specific techniques or using some specific antibodies against the viral particles. But even before the onset of the symptoms of the disease of the HIV, now by using PCR technique, if the virus is there, without the use of antibodies, without the appearance of the symptom, we can isolate the genome of the virus, and it can be amplified by using PCR, and then it can be detected. So before the disease, one can say that infection is going to happen or not. So it can be also used for the detection of viral diseases. In addition to that, we have studied that some specific type of PCR, suppose multiplex PCR, it can also be used to study different genetic diseases. I quoted the example of Duchenne muscular dystrophy. So this muscular dystrophy, it is caused by mutations in the exons of that gene, and the mutations in the exons, it can be detected by using multiplex PCR. So this is one of the example of the disease that can be detected by using multiplex PCR. In addition to that, prenatal diagnosis can be made by using PCR by taking the tissue from the fetus that is developing in the uterus, and even before implantation of the zygote, and this technique, it may be called as pre-implantation genetic diagnosis, or commonly as PGD, that if certain couples, they are having the experience of abnormal child, then they can opt this technique of PCR for the detection of genetic disorders in the zygote or at the zygote level as well. So in this way, PCR, it has been used in the medical sciences, not only in the detection of some genetic disorder, as well as in the detection of different microorganisms that can cause different diseases.